So next I want to introduce SHASHI AMUR from the FDA's Biomarker Qualification Program. All the speaker bios are in the packet, so we won't belabor the many qualifications of all of our fabulous speakers.
And first of all, I'd like to thank Dr. Wagner for inviting me to give a talk here and at the workshop here today. My title was Opportunities to Develop Meaningful Biomarkers, and I kept it the way it was, but I'm from CDER FDA, so to me, meaningful biomarkers relate to drug development. So today I would like to talk about two pathways that we have available to integrate biomarkers in drug development at FDA, one of which is biomarker qualification, and I'll talk more about that, and I'll give you an example of a recently qualified biomarker, total kidney volume, and then end with some take-home points. One question that's often asked is, which path do I choose? So it depends on the objective. If the objective is to use the biomarker in a single drug development program, your choice should be to go with acceptance through the IND, NDA, BLA submissions, and that's the drug approval pathway. And here, basically, a sponsor meets with the review division, talks to them about the rationale for using the biomarker, and then the biomarker gets accepted into the drug development program. Here, the risk and resources, of course, the burden is on that one sponsor. And once the biomarker is accepted in the drug development program, you might find the information embedded in different sections of drug labels, sometimes in reviews. So depending on the type of biomarker, the way it was used, that's how the information will be. If the objective, on the other hand, is to establish the biomarker for use in multiple drug development programs, your path should be biomarker qualification. Anybody, any individual or group can send in a submission for biomarker qualification. Oftentimes, we see submissions coming from consortia. And the risk and resources here are shared between the consortia members. And once qualified, the biomarker qualification recommendations become a draft guidance, and they become available on the FDA webpage. Here's the definition of biomarker qualification. It's a conclusion that within a carefully and specifically stated context of use, the biomarker has been demonstrated to reliably support a specified manner of interpretation and application in drug development. The term context of use comes, so that makes it different from the so-called validation. Context of use describes the manner and purpose of use for the biomarker in drug development. So when the biomarker is qualified, it's qualified for that specific context of use. This is a very important concept because it drives the level of evidence needed, and the level of evidence then drives the qualification process itself. We don't have evidentiary standards currently for the different types of biomarkers, and each type of biomarker can have multiple contexts of use, so the evidentiary standards are likely to be different for those contexts of use. So those are under development. We are working with multiple stakeholders, and through workshops and such, we hope to come up with evidentiary standards. In the meantime, here are some considerations for biomarker qualification. It's important to think about what's the type of biomarker you have and the context of use of the biomarker, as I mentioned already. The biological rationale is a nice to have. It increases your confidence in the biomarker, but there are proteomic signatures, genomic signatures, microRNAs, where the biological rationale may be a challenge. The relationship between the biomarker and the clinical endpoint or outcome are very important considerations. Thank you, Mark. You already covered the assay considerations, but I don't have to go into that. But we do look for an analytically validated assay so that we can trust the biomarker data. The type of data, many different types of data are acceptable in biomarker qualification, retrospective data, prospective data, et cetera. But again, reproducibility is very important. We have a learn and confirm paradigm. So you need a test data set and a confirmatory data set. Sometimes you can have one large data set that can be divided into two subsets prospectively. A prospective analysis plan, statistical analysis plan, is another requirement for biomarker qualification to take the bias out of the equation. This is a complicated graph or cartoon, but I want you to concentrate on these three things. So these are the three stages in biomarker qualification: initiation, consultation and advice, and review stage. The initiation stage corresponds to receiving the letter of intent and reviewing it, and the consultation and advice is with briefing documents and reviewing them, and the final qualification package is received in the review stage. Some time lines are indicated based on our experience, but we have streamlined the process some, and we continue to streamline the process to make it more efficient and more fast. And then wanted to tell you that there is something that happens even after the qualification recommendations are arrived at. We have to draft the guidance, then we have to clear the guidance. It takes a little bit of time there. So wanted to mention that. And the timeline, which is shown here, corresponds to only FDA time. The submitter time is extra. And submitters, of course, need time to generate data, analyze data, submit the data, and so on. We have so far qualified 13 unique biomarkers in 6 different submissions. First 3 submissions were on non-clinical safety biomarkers, and the next 3 are on clinical biomarkers. And I would like to give you an example of one of them, which is qualification of total kidney volume in autosomal dominant polycystic kidney disease. This was a submission from the Polycystic Kidney Disease Outcomes Consortium, and it was a joint EMA-FDA submission, and we worked very actively, collaboratively with EMA in this process. We had several meetings, and we had scientific exchanges, and comments and documents, all those we exchanged in this process. The approach of the consortium was, first of all, to standardize the data, which was something, Mark, you brought up in your talk. It's very important because you get data from registry studies and others. They're not identical, and they have to have the same data elements. So they went with the CDER standards. Apparently, it took them more than a year, 2 to 3 years, to get all the standards and the terminology and aggregate the data in order to use it. So basically, they took data from 3 patient registries and 2 longitudinal cohort studies and combined those. And then they used TKV, or total kidney volume, as a biomarker, and developed a disease progression model and sent all the packages, the LOI, the letter of intent, the briefing document, and finally the qualification package. The objective was clinical trial enrichment in ADPKD, and it was meant for use in all stages of drug development, such clinical stages, including proof of concept, dose ranging, and confirmatory clinical trials. The proposed context of use by the submitter is given here. Baseline TKV to be used as a prognostic biomarker in combination with patient age to help identify ADPKD patients who are at greatest risk in advancing the course of their disease, and the clinical endpoints, clinically meaningful outcomes they wanted to consider are 30% worsening of eGFR, 57% worsening of eGFR, and ESRD. I don't believe a lot of you know how much work goes in from our FDA reviewers. So I wanted to highlight that. We have a review team. We call it the BQRT, or the Biomarker Qualification Review Team. And this has reviewers with all the appropriate expertise. And they conducted a lot of additional analysis and also their own model development and cross-validation. The patient population that was used was trimmed down to limit to patients who represent populations likely to be enrolled in clinical trials. And also there were imaging modalities used, which were more than one. So there was a preference that FDA reviewers showed in choosing the MRI data over CT data over ultrasound data as the data became available. Also, FDA as well as EMA suggested addition of eGFR as a covariate. and inclusion of total kidney volume, in addition to patient age and eGFR, in the best fit model provided a modest improvement in predicting the risk of confirmed 30% decline in eGFR. And an external validation was also conducted by our reviewers to using a separate internal data set to verify the model. Though we were not qualifying the model, we were qualifying the biomarker. It was important to make sure the biomarker was working as the model predicted. There were too few ESRD and 57% decline in eGFR events, so meaning they were too few for meaningful analysis. I know I'm running out of time, but here is the draft guidance that we have. So basically, the proposed context of use is a little different from that which is qualified, because it only limits itself to 30% decline in the eGFR, and also it adds baseline eGFR as an enrichment factor to be used for TKV to be used in combination with age and eGFR. You can find the details at the web page. You can also find the executive summary and reviews at the web page, individual reviews. So one last slide before the red light is really distracting me. So here's the last but one slide. So FDA has launched some initiatives recognizing that qualification, biomarker qualification is a long process. People run out of time, resources, and other patients included. So the initiatives are critical path innovation meeting that helps with discussions on exploratory biomarkers, including in addition to other initiatives, and letter of support to show that to shine light on some promising biomarkers that to encourage biomarker development. These two are not part of the qualification program or process, but they help in that process. There's also another initiative I wanted to touch upon, that's the limited context of use qualification. If you narrow your context of use, the chances are you'll need less data to qualify the biomarker, which can be expanded later on. So that's all I have. Here's a summary. There are two paths, and biomarker qualification is a voluntary process, and it's meant to be used in multiple drug development programs, these biomarkers. And once qualified, this can be used by drug development in other applications without re-review for the qualified context of use. And new FDA initiatives hopefully can be utilized as early goalposts in biomarker development. Please talk to us early. Thank you.